evening everyone. I am Tania, a student of BSc first semester of Durgapur Government College. I am from Chemistry Department. So today I will discuss about the bioactivity of epicatechin and salicylic acid which I have prepared through computation. Okay, these are the contents of my project. Firstly, I will give the introduction of my project. Then we will get to know about what is bioactivity, the bioactive compound that I have chosen, and the reason behind it, objective of my project, the methodology that I have used, my observation, conclusion, and lastly I will give the references. Introduction. I have used computational method to know about the bioactivity of my chosen compound. So firstly, I will like to give a basic introduction about bioactivity through computation. Bioactivity through computation harnesses computational methods to understand and predict how molecules interact with biological systems. This interdisciplinary field accelerates drug discovery and molecular design by analyzing molecular structures and their biological effect using algorithms and simulations. What is bioactivity? As my project is about bioactivity, so first we should understand what is bioactivity. Bioactivity generally refers to the capacity of a material or substance to elicit a biological response when in contact with living tissues or organisms. This could include actions such as promoting cell growth, inhibiting bacterial growth, or including specific psychological responses. Basically, bioactivity refers to the ability of a substance, usually chemical compound, to produce specific biological effect or response in living organisms. These are the two biological bioactive compound that I have chosen. First one is epicatechin. This is the structure and the UPAC name of epicatechin. Epicatechin is basically a flavonoid compound which is found in certain plants and it have antioxidant and anti-inflammatory properties. Second one is salicylic acid. Salicylic acid is a beta hydroxy acid and commonly used in skincare products. Why I have chosen these compounds? So first one is epicatechin. I have chosen epicatechin for my project because it is the primary ingredient in chocolate and contribute to its adhesive properties. Second one is salicylic acid. I have selected salicylic acid for my project because it is the common ingredient in many skincare products. Now, objective of my project. We have used ChemDraw and Pass Online to analyze the bioactivity of chosen compounds. We have done the literature survey to this compound and explained them in this project. We have found the following useful bioactivities of the compound. So, for epicatechin, those are UGT2B substrate, phosphatase inhibitor, antiviral, apoptosis agonist, and reductant. For salicylic acid, dihydro-L-gluconate decarboxylase inhibitor, glutathione thioesterase inhibitor, sugar phosphatase inhibitor, aryl acetonitrile inhibitor, and lastly alkane 1 monoxygenase inhibitor. There is some problem in this slide, so I am requesting everyone just to ignore it. Okay, these are now methodology. We have used ChemDraw to draw the chemical structure of our chosen compounds and save them as MDL mol file. Next, we have registered ourselves on the Way to Drug Pass Online website and predicted the bioactivity of chosen compounds. Lastly, we have done a literature survey to explain the bioactivity of the chosen compounds and explain it in detail in the next section. Let's understand the methodology with the help of figures. Firstly, we have drawn the chemical structures in the ChemDraw application and saved them as mol file. Then we have registered ourselves in Pass Online website and predicted the bioactivity of our chosen compound. And lastly, we have searched the bioactivity on Google and stored the information. These are the observation of epicatechin and salicylic acid. These are the main findings of epicatechin. First is UGT2B substrate. So what is substrate? Substrate is like the raw material that gets changed by enzyme during chemical reaction. And UGT2B is a family of enzymes. So a UGT2B substrate is simply a substrate that gets affected by UGT2B enzymes. When epicatechin interacts with UGT2B, it gets transformed into other forms that our body can easily get rid of. So epicatechin can be considered a substrate for UGT2B because it undergoes a chemical change when it interacts with the enzyme. Next is phosphatase inhibitor. An inhibitor is a substance that can prevent or slow down enzyme activities. A phosphatase inhibitor is a substance that specifically inhibits the activity of phosphatase enzymes. Epicatechin has been studied for its potential bioactivity as phosphatase inhibitor. Research suggests that epicatechin may inhibit certain phosphatase enzymes, particularly protein tyrosine phosphatase, which play key roles in cell signaling and disease process. Antiviral. Antiviral refers to substances that inhibit the growth and replication of viruses within the body. They are used to treat viral infections. Now, epicatechin have antiviral properties. It prevents viral replication. Its activity against specific virus may vary, making it a promising candidate for further research in antiviral drug development. Next is apoptosis agonist. Apoptosis is a natural process where cells are programmed to self-destruct when they are old, damaged, or no longer needed. An apoptosis agonist is a substance or molecule that promotes the process of apoptosis in cells. The bioactivity of epicatechin as an apoptosis agonist involves its ability to promote the process of programmed cell death in certain cells. Reductant. The substance that loses electron and hereby oxidated to a higher valency state is called reductant. Epicatechin acts as reductant by donating electron to other molecules, thereby reducing them. Next is salicylic acid. So dihydroL-gluconate decarboxylate inhibitor. Salicylic acid has been studied for its bioactivity as a dihydroL-gluconate decarboxylate inhibitor. Showing promising potential in this role, this inhibition can have implication in various biological processes, including plant defense and mechanisms and medicinal application. Next is glutathione thioesterase inhibitor. Salicylic acid has shown potential as a glutathione thioesterase inhibitor, suggesting its ability to block the activity of this enzyme. This inhibition could have important implication in biological processes, but more research is needed to fully understand its effect. Next is sugar phosphatase inhibitor. Salicylic acid exhibits bioactivity as a sugar phosphatase inhibitor, impacting sugar metabolism and signaling pathways within biological systems. Arylacid nitrilase inhibitor. Salicylic acid has emerged as a promising inhibitor of arylacetonitrylase, an enzyme involved in various biochemical pathways. Its bioactivity in inhibiting arylacetonitrylase suggests its potential therapeutic application in modulating these pathways for various biological and medicinal purposes. And the last one is alkane 1 monoxidase inhibitor. Salicylic acid demonstrates notable bioactivity as an inhibitor of alkane 1 monoxidase and enzyme critical in alkane metabolism. This inhibition suggests potential application in environmental remediation and industrial processes reliant on alkane degradation pathways. Conclusion. The computational study of the bioactivity of epicatechin and salicylic acid offers valuable insights into their potential pharmacological effects. By leveraging computational method, we have elucidated the molecular interaction and possible mechanisms of action. Further experimental validation is warranted to corroborate these findings and pave the way for the development of novel pharmaceutical intervention harnessing the bioactive properties of these compounds. So these are the references that I have used, and lastly I want to thank everyone. Thank you all.